Well, thank you for letting me substitute for Gary. We actually, he stayed in California to close a $35 million round last night, which we announced this morning, so very excited. We are Advanced Cell Technology. We are a publicly traded company, so I do need to put up the forward-looking statement. I'm going to start by talking very quickly about the two platforms that we're using in terms of our cell therapy programs. The first is an embryonic stem cell platform. We actually derive our embryonic stem cells in a fundamentally different way than Jamie Thompson and others in the field had done this. We actually developed a technique that allows us to generate embryonic stem cells without harming or destroying embryos. And what we did is we looked at what was going on in the IVF clinics with regard to being able to do pre-implantation genetic diagnostics on embryos that were created in vitro, and realized that the single cell biopsy that was being used for that genetic testing on those embryos could also be used to generate embryonic stem cell lines. And so in 2005, we generated about a dozen lines. Those lines actually will carry us forward through all of the therapeutic programs that we can imagine. You know, each one of these lines, by definition, is self-replicating, self-sustaining. So, you know, the expectation is, we could make 1 trillion doses of RPE cells and as many doses of mesenchymal stem cells and some of the other things we'll talk about today from these lines. We have one line that has already gone through and become a GMP-compliant master stem cell bank that we're using in our current trials, and we're doing the same thing now with multiple other lines that will not only use our cells but make available to potential collaborators in other spaces, such as those that are generating motor neurons or dopaminergic neurons and the like. We also have a program in IPS, induced pluripotency stem cells. We actually used to call this de-differentiation. It's a field that we've been working in for quite some period of time. Well before Yamanaka and his group published on the 4 factors, we had been playing around with factors like OCT4 in terms of inducing pluripotency. So we have continued to move forward with that as a platform, as well as I asked the leading question earlier of Brock and Ilona and others. I was kind of curious to see whether the view was different. Hopefully, actually, we'll have a little surprise for you today. So I am going to talk about 3 programs. We have a lot of other things in the pipeline, but I'm going to start with the clinical program that's actually already in human trials right now, and that's our RPE clinical program. Let me give you a quick biology lesson. If you look at the retina, the back of the eye, you see a complex microarchitecture that exists. The light actually comes in this diagram. I flipped it 90 degrees, and the light comes through the top. You have the nerve fiber layer. Under that are the ganglia and bipolar nerve cells. Below that are the photoreceptors. Underneath that is the retinal pigment epithelial, which I just shorthand as RPE layer, the Brooks membrane, and then the choroidal space below it, which is the vascularization underneath. And it turns out that in that microarchitecture, the RPE cells play a very critical role in both maintaining the structural integrity of the space as well as the health of the nerve cells above them. And part of that is the fact is that this subretinal space does not see blood flow. So all nutrient and waste exchange is mediated by the RPE layer. So the RPE layer provides critical nutrients, growth factors, even water to the photoreceptor space above it. It's also a part of the photopigment cycle. So the photoreceptors use 11-cis-retinal as part of the mechanism to convert light into a nerve conductive signal. That produces an all-trans-retinal, which the RPE layer then recycles back to 11-cis-retinal. So it's part of, it maintains this kind of metabolic cycle that's required ultimately for sight. The other thing that it does is it moves out the waste. It moves out the other metabolites of the photoreceptor pathway, so A2E, for example, is one. The photoreceptors also routinely shed what are called the outer segments over time. That cell debris is phagocytosed by the RPE layer, and then ultimately the waste is secreted into the choroidal blood flow below it. So it has this detoxification role. And then the other thing that it does is it actually it secretes and maintains the Brooks membrane below it. That Brooks membrane is the natural angiogenic barrier that prevents vascularization in the back of the eye. And then the last piece of it is it does absorb stray light. It is a pigmented cell. Like the melanocytes in your skin, these cells are dark. When you look into someone's pupil and it's black, you're seeing the RPE layer in the back of their eye. So it turns out that these cells, have fundamental roles, not just in normal tissue, but it's the failure of these cells that often precedes a number of different macular degenerative disorders. The ones that we're all familiar with, of course, is age-related macular degeneration. There's dry AMD, which makes up about 90% of all AMD patients, and wet AMD, which represents about 10%. In the case of dry AMD, you see the loss of the RPE cell layer that then is followed by a buildup of this toxic waste, drusen, the A2E metabolites, the photoreceptors, and that results ultimately in degeneration of the photoreceptor layer. 90% of all wet AMD patients progress from dry AMD, and so the disease actually is a little more advanced. As you lose more and more of the RPE cells, not only are you seeing degeneration of the photoreceptor layer, but you're seeing loss of the Bruch's membrane. And once you lose the integrity of the Bruch's membrane, that allows the vascular underneath it to invade the back of the eye and ultimately disrupt the structure in the back of the eye, and that's what we call wet AMD. So again, it was a very discrete cell population. It turns out it's a fully differentiated cell that you could actually think about targeting for therapy. If you could replace the cells that had died in the back of the eye, if you could resurface that Bruch's membrane, you potentially had the opportunity to be able to recapitulate the function of the RPE layer and either stave off the progression of disease, or in some cases maybe be able to restore some of the visual acuity where the photoreceptors were still there, but had just atrophied or lost the ability as a consequence to the lack of being able to recycle those photopigments. There are a lot of reasons to think about the RPE cell as really an ideal cell therapy product. They're easy to identify. As I mentioned, they're pigmented. It turns out that pigmentation combined with PCR gives us a sensitivity in our release criteria of being able to detect 1 in 20 million cells in terms of contamination with any undifferentiated cell. And this was from the FDA's perspective, this was actually significant to them. And we're talking about small doses. We're talking about injecting 200,000 cells or less into the subretinal space of these patients. The back of the eye, again, because of the Bruch's membrane, in the case of dry AMD, is generally considered to be immunoprivileged. So the expectation was that, light course of immunosuppression at most, we're actually aiming eventually in earlier stage patients to potentially get to a place where we can use no immunosuppression. And then finally, ease of administration. At least in the case of the dissociated cells that we're working with, we were able to consider a therapy that used an off-the-shelf standard cannula injection system. We didn't have to design a new injection system that also required separate regulatory approval. We were able to use something that's already routinely used by the retinal vitreal surgeon community. And again, we talked about dry AMD, but the fact of the matter is just there's many, many different forms of macular degeneration that are impacted by the loss of these RPE cells. So there were many different therapeutic targets that we could consider. Dry AMD, of course, is, that's the holy grail in ophthalmology. It remains really one of the biggest unmet medical needs worldwide. There are 30 million patients just between the US and Europe alone today that have some form of macular degeneration. Again, about 90% of them have dry AMD. It is the leading cause of blindness in people over the age of 65, and currently there are no approved therapies that are effective in treating this. For the most part, people are told to take vitamins. Our target in our clinical trial, obviously, as a safety study, we'd be looking at the late-stage patients, but our end goal is to be able to use these cells to resurface and replace missing RPE cells at much earlier stages with the hope and expectation that we can actually halt the progression of this disease altogether. So with that in hand, we began gearing up towards clinical trials. And one of the first things that we did was to work through a whole GMP manufacturing process that begins with one of these embryonic stem cell lines that we created in 2005. We created a master stem cell bank. We optimized this process for being able to manufacture these cells. We have a 1,000 square foot clean room, which is probably half the size of just this portion of the room, and in that clean room we can manufacture almost 500,000 doses a year with just a single 8-hour shift. And part of that is because they're small doses. A single 6-well plate will make almost 50 doses of cells. What's beautiful about this is that, particularly for the big life science companies that are afraid of cell therapies, is that this looks so much like an antibody or a protein therapeutic. In fact, there are many aspects to this that are easier from a manufacturing process than even monoclonal antibodies. We're talking about cells that are cryopreservable, so we can centrally manufacture them as a consequence to using this allogeneic source of embryonic stem cells. We can ship them worldwide. We ship them already to Europe for our clinical trials there. They're easily brought back up at the clinical trial site, and then, as I said, injected using a standard system. So really a great first product opportunity for any company when we started thinking about where you might go with a cell therapy product at least from an embryonic stem cell source. Went through extensive characterization of these cells, again, as part of our IND packages. We do everything from regular morphological assessment of these cells and karyotyping to sterility testing. We're routinely pulling samples of both the master stem cell bank, the working cell banks, and the clinical banks of RPE cells that we have. Along the way, we developed a number of potency assays in order to be able to get even tighter control over the manufacturing process. One of those potency assays that we've developed allows us to quantitate the phagocytic activity of these cells. Again, from the perspective of the activity that's required in the back of the eye to clear up that cellular debris. This is a function that we wanted to make sure that we had the ability from batch to batch to be able to test. One of the other things that was surprising to us was that the degree of pigmentation mattered, and it turned out to matter in a not so obvious way up front. So we actually did develop a quantitative assay for measuring melanin content in these cells over time to decide when we would repasture the cells and when we would harvest them for cryopreservation. We also did extensive animal studies. We did all kinds of tumorigenicity studies. I threw up just a couple of slides here to give you a sense of some of the anatomical and functional data that we had that we presented to the FDA. In the upper left-hand set of slides, this is a mouse model for Stargardt's disease, which is a form of juvenile macular degeneration. And what we were able to show when we injected human RPE cells into this mouse model is that they recapitulated, they resurfaced the RPE layer that had been lost. And they did so not only to form a monolayer, so you didn't see clumps of cells, but you can actually see in this panel over on the right-hand side, you see the mouse RPE cell is not labeled. The human cell is GFP labeled, so it lights up green. You can see that the human cells actually sit down adjacent to the mouse cells and then form tight junctions with the mouse RPE cells that remain behind. So again, anatomical evidence that these cells were able to get to the right place by injection and then ultimately recapitulate the appropriate structure. Functional data, this is an example of it on the bottom. This is the rat model for 10 minutes. Oh, I have to go faster. These animals had maintained their photoreceptor layer when the untreated animals would lose their photoreceptor layer within 6 months. These animals had visual acuity about 70% that of the wild type animal. These are the clinical trials study design. We have a Stargardt's trial going in the US, a Stargardt's trial going in Europe. We also have a dry AMD trial going in the US. They're ascending dosage formulations, 50, 100, 150, 200,000 cells. Endpoints, because it's a safety study, are pretty safe. This is pretty straightforward. I'll let you read these online, but we do have the ability, because we can image the back of the eye with OCT and other techniques, to be able to look for evidence of engraftment, and as well as detect visual acuity changes in these patients. Dry AMD endpoints are substantially the same. We were able to recruit some of the top retinal vitreal surgeons and some of the top eye hospitals in the world to participate in the trials here in the US and in the UK. We have Jules Stein, Wills Eye, Bascom Palmer, Mass Eye in here, Moorfields Eye Hospital in London, Edinburgh Royal Infirmary, obviously in Edinburgh, and Bascom Palmer Eye down in Miami that are participating in the trials. This is a quick video. This is our first dry AMD patient. We treated this patient in July of 2011. You can see the big area of atrophy in this patient. We do a vitrectomy followed by a PVD induction. This is now the injection. This is 150 microliters of cells, so about the volume of the tip of your pen. You can see that in the subretinal space, that fluid fills up. It looks like a little bubble under a piece of wallpaper. When this pulls back, you'll see where this injection was made. It was made right on the edge of this patient's atrophic retina, and then what looked like some normal retina next to it. And then the last step is an air fluid exchange. You'll see air start to pump into the eye. This puts pressure on the bleb. The bleb resorbs over a period of about an hour or two. This is a day patient surgery. There's no general anesthesia. This is sedation, much like LASIX. I trimmed 15 seconds total out of this video, so you actually are seeing the vast majority of this surgery in real time. That's how quick this is. The Lancet actually took, I think, an unprecedented move in terms of having us publish at 4 months the data from our first 2 patients. We treated not only that dry AMD patient in July of 2011, but also a Stargardt's patient as well. This was right on the heels of when Geron had kind of unceremoniously pulled out of its embryonic stem cell program. So I think they don't make their reputation on publishing papers on 4 months worth of data on 2 patients from 2 different trials, but yet this was something that they wanted to get out there. Bottom line is that in the paper, what we published is that we saw no adverse events. We saw that the evidence that the cells had persisted over time, and I think was a surprise to us, is we actually were measuring improvements in visual acuity in these patients. So where are we today? So the original visual acuity measurements in both of the Stargardt's patients and the dry AMD patients, we moved them in ways that were kind of hard to think were just happenstance. And these improvements in visual acuity have been maintained. Our Stargardt's patient went from hand motion only, in other words, she couldn't count fingers on her hand, to reading three lines on a chart. And 14 months later, this says 12 months, I just saw her 14-month data, she's actually slightly improved since then. So the effect has persisted. These patients have been weaned off of their immunosuppressants for more than a year, so it's not an immunosuppression issue, which some people will ask about, but it's something that seems to be the consequence to the persistence of the cells, understandably still very early stage. The next longest patient we have is our first patient in the UK for Stargardt's. Same thing, we saw an improvement in visual acuity. We have 11 patients treated now. 9 of them have measurable changes in visual acuity. This is happening at 3 different clinical trial sites, and it's happening with multiple follow-ups. These patients, by the time you get to 14 months, they've been seen 25 to 30 times by the team at UCLA. That's actually doing these measurements. But from a safety perspective, 7 SMD patients, 7 Stargardt's patients treated. All of them are doing well. No evidence of inflammation or infiltration, no ectopic tissue, no tumor, no evidence of retinal detachment. Likewise, we have 4 dry AMD patients into the trial. Same results from a safety perspective. I'm going to actually skip through this. From a price justification, we think that this is a therapeutic that ought to price somewhere, conservatively between $5,000 to $10,000 a dose. You hit 1% market penetration in a 50 million patient population worldwide, and you're looking at a $5 to $10 billion drug. I highlighted that in blue for Dean, who unfortunately I guess has already left. So there we go. Moving right along, MSC program. This is something we haven't talked a lot about publicly. We heard the folks from Mesoblast talk about this earlier, so I will skip a lot of this. The beauty of mesenchymal stem cells is you can use them systemically without having to HLA match them between the donor and the recipient. They are on their own immunoprivileged. So it allows you to consider having centralized banks. Huge potential in things like autoimmune and inflammatory disorders, and there's already a track record out there with over 200 clinical trials underway right now. And so you'd ask, why would we think about getting involved in this? Well, we still think that there are some issues with adult sources of mesenchymal stem cells, and really it all comes down to the replicative capacity problem. You can take an adult source of mesenchymal stem cells, whether it be bone marrow or whether it be fat, and you can only get a couple of passages out of it before you see these cells start to undergo replicative senescence. And that has implications on being able to create banks. It means you're constantly going back to donors, and even if you've worked out a way to come up with donors regularly, if you could imagine a business where there are millions of doses of this a year, we're talking about tens of thousands of donors are going to be required as sources. And so we looked at this and said, is there an opportunity using a pluripotent stem cell source to be able to create a centralized manufacturing source of whether embryonic stem cells or iPS cells that would allow you to make mesenchymal stem cells that were at least competitive with adult sources? And the answer is yes. We're actually able to make tremendous quantities of mesenchymal stem cells from ES sources. It turns out that these cells are very potent. They actually have qualities that make them more similar to fetal MSCs than to adult MSCs. And you avoid the replicative senescence problem. These cells will actually hold aside the fact that you have a ready source of a master stem cell bank that you never need to go back to a donor for. These cells will go more than 50 passages before you see any issue of replicative senescence, as opposed to 2 to 4 in the case of adult derived. So it has a big impacts with respect to manufacturing. In our animal models, we saw something that was also very intriguing to us, and that is that we were seeing that the potency of the early passage MSCs that we're making from mesenchymal stem cells were far more potent than later passage ES-derived MSCs or than the bone marrow-derived MSCs, and by pretty substantial margins. If you take a passage 30 to 40 MSC that you derive from an embryonic stem cell line, it has a potency about on par with bone marrow-derived MSCs. Between passage 10 and 20, there is a profound difference in the potency of these cells. They also will persist in vivo longer than the corresponding bone marrow. So from our perspective, it looks like it gives you, not only solves part of the banking issue and the sourcing issue, but potentially gets you to broader ranges of diseases based on potency, allows you to go to lower doses to avoid potential, improve some of the safety profile for these cells, and then perhaps be able to go longer between injections. Lots of potential applications. And the last one I'm going to get to is we have a vascular program. We start with a hemangioblast. We can now make this from either ES cells or IPS cells. The hemangioblast is the precursor to the entire hematopoietic lineage as well as the vascular endothelial. We are now making enough platelets from megakaryocytes that we're deriving through this pathway that we're now starting to look at a pre-IND meeting with the FDA on the near-term calendar, proposing to file INDs for both ES-derived platelets as well as IPS-derived platelets. This just shows you the ability of the embryonic stem cell-derived platelets to participate in clot formation and clot retraction, and we actually have some in vivo data as well. So this is where we are. Thank you. That was as fast as I could talk.
Thanks, Matthew. I still want to give him a chance to answer questions, so if you have any questions, please go ahead.
Thanks. It's very nice stuff. On OCT,
Yep.
especially for the dry AMD patients, are drusen regressing or absorbed?
So I'm afraid I don't know the answer from OCT. We actually have a meeting of all the clinicians in a couple of weeks that have been involved in this, and I'll probably get a chance to see a little bit more of the data. From the business development standpoint, I kind of get the anecdotal. What I can tell you from OCT, what I've seen is that in the case of the Stargardt's patient, we were able to show that the injected cells get to the Brooks membrane and form a monolayer. With regard to drusen, again, this is anecdotal, that's come back to me, is that in our most recent Stargardt's patient, where she hasn't seen a change in visual acuity, they think they're seeing changes in autofluorescence. It's not well controlled, and that's one of the conversations we're having in a couple of weeks. There's an expert here at Mass Eye and Ear on quantitative autofluorescence, but that's the type of thing that I think we want to start looking at function, even if we can't see the cells yet by OCT or see changes in visual acuity in these late stage patients. Again, these are patients that are all legally blind in the eyes that we treat them in.
You mentioned the use of markers for the cells and release criteria?
Oh, no, what I was saying is that one of the advantage of pigmentation is that it turns out to make it the release criteria for demonstrating lack of contamination with undifferentiated cells, in particular embryonic stem cells, a lot more robust than PCR was. No, yeah, so PCR is a secondary release criteria for us in terms of looking at markers for RPE versus markers of embryonic stem cells.
Did you find the markers are context dependent? How stable were the markers in the populations?
I don't know the answer to that. I guess I would answer it with saying stable enough for the FDA. And the MHRA. I mean, this wasn't just the FDA that's looked at this. The UK's MHRA has looked at it as well. We may have that in the Lancet article. I'm afraid I don't know the answer off the top of my head, though.
Any other questions? Was the ES-derived MSC. So one thing that made bone marrow MSCs very successful in the clinic is basically the history of them. That they were, well-known, well-established, they were given to patients long before, as part of bone marrow even. So I'm wondering how regulators view ES-derived MSCs.
We'll find that out.
Yeah,
I mean, the answer is no one knows yet. I think that, I mean, obviously there's going to be an extra layer of oversight on this, right? I mean, the issue is going to be demonstrating that there's no contamination with an embryonic stem cell or an iPS cell in that. You know, we knew that was significant in getting through the FDA on the RPE side. It's going to be as significant, if not more significant here. And I think the other piece is they're going to want to see some history with, as I pointed out, these look more like fetal MSCs than adult MSCs. Is the safety studies are going to have to reflect that too. That there's no, there's nothing different in terms of the pluripotency or the path that these cells could go along. That would be different from adult. I think we get to ride the coattails of adults in a lot of ways, but I think there's extra work we know that we're working through.
Okay, great, thanks.
Okay, thanks guys.